Good evening, people. Hi, Bowen. Hello. Can you hear me clearly? Lamai, can you hear me clearly? You can hear me, ne? Yes. Good evening. Good evening. Good evening. Everything fine, ne? Right? You can hear the sound of rain from the background. How many of you came for the class today? How many of you came for the class? Uh, how many of you came for the class? One person, two, Rifka. Only two, ne? Others didn't come. Only two, Samadhi and Rufaika. Uh, there are some more, ne? Right? Okay, people. Things. Any Kurunagala people here? Kurunagala students in the class? Kurunagala guys? Kurunagala people? Who's Kurunagala? Kurunagala? Hello? Anyone from Kurunagala class? Kurunagala? Anyone? No one? Nobody? Right, people. So I'm still with the same dress. Just now came home. I had a small class, speed class for the 2027. Finished it. Heavy rain, eh? Heavy rainfall. Looks like it's developing into a cyclone again, eh? Eh? Looks like it's developing into a cyclone. So in Sri Lanka, somewhat, eh? Cyclone. We don't know, but looks like some heavy rainfall can be expected in the coming short time period, right? So stay alert, right, people. We have to discuss paper number 13. Tomorrow's Kurunagal class I'll take online. If any Kurunagal student in the class, please raise your hand up. Right, we'll have the class tomorrow morning online. I'll give you the link. I'll be teaching you all live. So we have to do the same lesson, excretion lesson, right? Other parts I think we are parallel with the Candy class, right? Lamai. Okay. Now today's discussion will go with paper number. We'll go with paper number 13, Lamai. Paper number 13. Paper number 13. Right? Hello? Paper number 13, okay? So we'll take the paper number 13. No, we'll go with paper number 14. Paper number 14. Paper 14, Lamai. Ooh. Paper number 14. Okay, scheme 13. Where is that? Okay, just give me 1 second. Paper number 14, Paper number 14, paper 14, paper 14. Right, paper number 14. Right, people, this is the paper. We'll go with the 14th paper. 14th paper, 13th paper I discuss partly, but I have given you the marking scheme. I think you can manage with that. So that's not a difficult paper anyway. We'll go with paper number 14, my dear students. Did you answer, all of you, the paper? Right, first question. The ability of organisms to change over time as a result of genetic modification. What? Yeah. The ability of organisms to change over time as a result of genetic modification. The ability of organisms to change over as a result. Evolution. So we'll take evolution as the answer. Evolution as the answer. Evolution. Evolution is the answer. Question number 2. Which of the following polymer of ketose type sugar, polymer of ketose type of sugar? Are you going to tell me the answer? Are you going to tell me the answer? So your background is noisy. Is it any extra noise? Any extra noise? Is it clear, people? Is it clear? It's raining. It's a metal roof here. Right? So it's a metal roof. Look at this metal roof. Oh my God. What did I do? Right. Question number 2. Which of the following polymer of ketose type sugar? Ketose type sugar, you can take inulin, because inulin is a fructose polymer. Fructose is ketose, ne? Fructose is ketose. Question number 3. Correct regarding cell wall. It's found only in plants and fungi. No, bacteria has cell wall too. Wrong statement. The first deposited cell wall during the cell division is secondary cell wall. No, it's the primary cell wall. Wrong. Secondary cell wall is found interior to the primary cell wall. Secondary cell wall found interior to the primary cell wall and plasma membrane. Wrong. It's actually present interior to the primary cell wall, exterior to the plasma membrane. Wrong statement. There are free spaces found in the cell wall where water can move freely. That's correct. Intercellular spaces are there. The chemical composition cannot vary even in the same species. No, within the same species you can see that the chemical composition of the cell wall can be variable. You know, some parts are going to be lignified, some plants are not lignified. Suberin can be there, cutin can be there. Cell wall composition is variable. So I am taking fourth one as the answer. I hope you got the correct answer too. Question number 4. Which of the following statement is correct regarding enzymes? Some enzymes alter the nature of the end products of reaction. Some enzymes alter nature of the end products. Wrong statement. You know, enzymes do not change the nature of the end products. Inhibitors bind with the enzymes reversibly by covalent bonds. Inhibitors bind with enzymes reversibly by covalent bonds. Now you know there are two types of inhibitors available, reversible and irreversible. I think we cannot agree with the second statement fully. Question number 3. ATP can act as allosteric inhibitor to some enzymes. ATP act as an allosteric inhibitor to some enzymes. ATP act as allosteric inhibitor. Oh, activator, people, inhibitor correct, ne? ATP act as allosteric inhibitor to some enzymes. Yes, we can agree with that. That is correct. ADP is allosteric activator, ATP is allosteric inhibitor. We agree with that. Some non-competitive inhibitors bind with the active site of the enzyme. Some non-competitive inhibitors bind with the active site of the enzyme. Non-competitive inhibitors are not binding with the active site, but they go and bind with the allosteric site. Wrong statement. The active site of the enzyme is made up with many amino acid. The active site of the enzyme is made up of many amino acid. No, we learn people few amino acids are available, not many. It can be 3 to 12, but not more than that. So therefore, we'll take third one as the answer. Are you agree with me, people? ATP can act as allosteric, that one, allosteric inhibitor. I'm taking that one as the answer. Next question is about photorespiration. Photorespiration question, my dear. It is the process of consuming carbon dioxide and releasing of oxygen. Wrong. Photorespiration is not consuming carbon dioxide and releasing oxygen. We know, people, it's something similar to cellular respiration. Something similar to cellular respiration that happens in plants, when the carbon dioxide level is too low. You learn that. And second one, take place only in C3 plants. Take place only in C3 plants. That is not correct. C4 plants happen repressed, but that doesn't mean it's completely stopped, neither repressed. So maybe some of the C4 plants it happened too. Question number 3, each time Rubisco reacts with carbon dioxide instead of oxygen, the plant makes 50% less PGA. Each time Rubisco reacts with carbon dioxide. Wrong. Not with carbon dioxide. Every time as it reacts with oxygen. Neither. Wrong statement. Carbon C4 plants largely repress photosynthesis. Perfect statement. That is correct. Photorespiration is not energy demanding. Wrong. Photorespiration needs energy. We got the answer anyway, people. Fourth one is the answer. Fourth is the answer, right? So the marking scheme answer is correct. Question number 6. An event occurring during prophase of mitosis is. An event occurring during prophase of mitosis is chromatin fibers get relaxed. Chromatin fibers get relaxed. An event occur during prophase of mitosis is chromatin fibers get relaxed. Prophase of mitosis is prophase chromatin fibers not getting relaxed, neither get condensed, neither people condense into chromosomes. Relaxing is a wrong word. Then centrosome move towards opposite poles of the cell. Centrosomes move towards the opposite pole. That is correct. Centrosomes are not centromeres, right? Centrosomes are a pair of centrioles. They move to opposite side. That is perfect. Correct statement. Second is okay. The nuclear envelope fragments. Nuclear envelope fragments during the prophase. Am I correct? Does that happen? What is the phase for the fragmentation of the nuclear envelope? Someone is going to tell me the answer. Pro-meta. That is correct. Tahira. Very good, Tahira. That is pro-metaphase. So we can't agree with that. And then the kinetoco microtubules move the chromosome back and forth. That also happen in the pro-metaphase. Wrong statement. Spindle microtubules get depolymerized. Spindle microtubules get depolymerized. That happens maybe in which phase? Depolymerization. Telophase. That is Sabik. Very good, Sabik. So we'll take people answer as the second one. Answer is second one. Centrosome move towards opposite pole. That's the answer. Maybe I feel like I should give you the answer after the discussion of the paper. Normally now I print the marking scheme on every Friday, right, and then hand it over to you on Saturday. Do you think people handing over the paper marking scheme should be done after the discussion? What do you feel about it? If you feel that's better, I can do it. Or is this way is okay, or should I delay it little bit and then give you the marking scheme after the discussion? How you feel it? How you feel it? Say is this method is better, or should I give it after the discussion? Tell me. Because you feel like I have answers. There is nothing to mark. Some people may be not joining because they have the marking scheme. How do you feel? Is this okay? Is this method okay? Someone you think says yes කියන මොකද්ද? What do you mean by yes? This is okay the. I feel like I should give it after, right? No comments on that. No one comments on that. Okay. Question number 7, my dear. Question number 7. You have to send me your chats, people, to the main chat line. People, I can't see the mains. I can't see how to obtain recording of the today's extra class. Recording after discussion. Some people say after discussion. This method is okay. 50-50. 50-50. Question number 7. Today's class among group. I'll put that to the group. Okay. Right. Question number 7, people. The Kelvin cycle of photosynthesis. Please give the marking scheme after the discussion. Now, a couple of people say to give it after the discussion. I feel like that better, after discussion marking scheme. We'll plan for that, so that I can collect your paper and then staple it, the marking scheme, and I can give it. Question number 7, the Kelvin cycle of photosynthesis. Kelvin cycle of photosynthesis. PEP carboxylase enzyme involving in carbon fixation first. PEP carboxylase Kelvin cycle. In the Kelvin cycle, PEP carboxylase involving in carbon fixation first. Can't be neither. PEP carboxylase enzyme. If it is a C4 plant, that's correct. C4 plant, correct. In the Kelvin cycle of photosynthesis, PEP carboxylase enzyme involving in carbon fixation first time. A Kelvin cycle PEP carboxylase does not involve. That is not correct. Kelvin cycle. Kelvin cycle. Kelvin cycle. So we'll take that is wrong. Glyceraldehyde 3-phosphate would be reduced. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. G3P, G3P would reduce wrong. 1,3-bisphosphoglycerate reduces. Not glyceraldehyde 3-phosphate. That is wrong. Remember the Kelvin cycle. Remember Kelvin cycle, Lamai. Kelvin cycle, Matakada. Remember Kelvin cycle, can you write the Kelvin cycle? RUBP. RUBP PGA 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate G3P. And then RUBP. 1 G3P goes out. G3P goes out. 5 G3P goes back. How many RUBP here, my dear? 3 RUBP. 3 carbon dioxide. 6 PGA. 6 1,3-bisphosphoglycerate. 6 G3P. 1 G3P goes out. 5 re-enters. And here we can see that ATP. ATP becomes ADP. 6 here 6 NADPH becomes 6 NADP plus. That's it. That's the process. Alright. So here what it says, glyceraldehyde 3-phosphate reduce. G3P don't reduce. Reduction happen here. Ne? These are the 3 steps, people. This is carboxylation, number 2 reduction, number 3 there. So here to here a reduction, not from here to there. So that is wrong. And then decarboxylation take place during reduction. No, reduction there is no decarboxylation. No decarboxylation here. Carboxylation occur. No decarboxylation. Wrong. Regeneration of RUBP need energy in the form of ATP. Regeneration of RUBP needs energy in the form of ATP. Correct. I forgot to mention it here. Here you can see some more ATP needed. Ne? ATP. ADP. Right. So that is needed. This happens there. Correct. And then regeneration of RBP needed, that is correct. Only NADPH needed for reduction. Only NADPH needed for reduction. Only NADPH needed for the reduction. Why it's wrong? Only NADPH needed for the reduction. Only NADPH needed for the reduction. Only NADPH needed for the reduction. Regeneration of RBP need energy in the form of ATP. Fourth one is okay, but then I have little doubt, people. Only NADPH needed for the reduction. This one is the answer. Only NADPH needed for the reduction. Reduction step 1,3-bisphosphoglycerate. I think resource book ke ATP mention kala tibunada. I think there was an error in the resource book. Resource book, there was an error in the resource book. Shall we check that one, people? Resource book, there was some mistake. I think resource book explains that it needs resource book says reduction needs ATP. Resource book says, I think that's the problem. Remember that? Can you hear me, Lamai? Then mukaddu error එකක් තිබුණානේ the resource book එකේ. Right, when regeneration, reduction phase. This one neither. Make an issue. PJ is converted to 1,3-bisphosphoglycerate by adding phosphate. 1,3-bisphosphoglycerate will be reduced to glyceraldehyde 3-phosphate through the step-by-step enzyme catalyzed reaction using NADPH and ATP from light reaction. Itna tama error this ATP story. That ATP. But anyway, now resource book even if it is wrong, people, we have to take like that. So anyway, people, the ATP does not involve here. We learn the process. ATP does not involving in this reaction, but the resource book says that. That's an error in the resource book. That's a very, very poor mistake, people. Right? Poor mistake. We can't argue all these things. Right? Anyway, so I'll take the best answer as the fourth one. Right? Fourth one is the best answer, dear. So we'll Right, people, we'll take the next page. We'll move to next one. We'll move to next page. Question number 8. Question number 8. Shall we? So please give marks. Okay. Parane kakne de. Ilanga page and question number 8, people. Important function of fermentation is to. Important function of fermentation is to synthesize glucose, production of ethyl alcohol, prevent oxidative phosphorylation, produce NADH, regenerate NAD plus. You know, people, the answer is this one, regenerating NAD plus. Regenerating NAD plus. That's the most important process which happens in anaerobic respiration or in a fermentation process. You learn, people, in lactic acid fermentation or even people in ethyl alcohol fermentation. You know, pyruvate, wait, we start with glucose, ne? Glucose becomes pyruvate and pyruvate transforms into lactate, lactic acid. Here we know NAD plus becomes NADH. So fermentation, the whole fermentation aim is to convert this NADH back into NAD plus so that it can maintain glycolysis. Neither ADP becomes ATP. Right? The whole purpose is that. So we are taking fifth one as the answer for that question. Question number 9. Question number 9. The three eras of Phanerozoic eon is correct chronological sequence. Phanerozoic, Paleozoic, Mesozoic, Cenozoic. Neither. So we will take, people, second one as the answer. Second is the answer. That's quite easy question. Ne? Paleozoic, Mesozoic and Cenozoic. Next one. Question number 10. Question number 10. Characteristics not possessed by an organism which has silica as a cell wall component. Silica is available as a cell wall component in diatoms. Diatoms, good. A character not possessed by an organism which contains silica, being marine as well as freshwater, correct. You see diatoms present in freshwater as well as in marine environment. The body comprises of two overlapping parts, that is also correct. Two shells, two halves are present in the diatom body. Being multicellular, wrong. Being multicellular, wrong. Character not possessed by an organism which has silica in the cell wall component, being multicellular, wrong. So multicellular, wrong. Multicellular, wrong. Being photosynthetic, correct. And then having chrysolaminar as storage food, correct. So now we have the answer, third one. I think marking scheme answer is wrong. Lamai, can you correct the marking scheme answer? The question number 10 answer is 3. 10 ke 3 ne lamai. 10 ke 5 kila dilatini, it has to be 3 ne. It has to be 3. Right. Question number 11, people. Characteristics of different phyla of seed plants are given below. Which pair is correct? Characteristics of seed plants are given below. Which pair is correct? Netophyta leaves look like those of the flowering plants. Netophyta leaves look like those of the flowering plants, correct. Eka Saria. That is Sari, neda. That is Sari. And Psychodophyta have flagellated sperms. Psychodophyta flagellated sperms, correct. Sari. Coniferophyta, 2 types of spores are 1 cone. Coniferophyta, 2 types of spores in 1 cone. No. Coniferophyta, 2 types of cones are there. Wrong statement. And then people, Anthophyta, some fruits have persistent floral parts. That is correct. Calyces, persistent floral parts, they don't fall. Correct. And Netophyta seed looks like seed of the angiosperm. Netophyta seed look like the seed. Fruits. Seed looks like seed. Seed looks like what? Seeds look like what? Netophyta seeds look like what? Look like what? Looks like what? Come on, my students. Come on, answer me. Seeds look like fruits. That is seed and wrong ned. Enam people ABD. ABD, third one is the answer. Question number 12. What is the true about fungus? What is the truth? What is the true? Correction needed. What is the truth about fungus? Oh, what is true? What is true about fungus? Zygomycota mycelium has incomplete septae. What is true about fungus? Zygomycota mycelium has incomplete septae. Zygomycota mycelium has incomplete septae. No septae neither. Zygomycota don't have septae. Wrong statement. Who's having septae? Ascomycota has septae. Basidiomycota has septae. Wrong statement. All fungi are absorptive and decomposers. All fungi are absorptive and decomposers. Wrong neither. Some of them are mutualistic. Some are mutualistic and symbiotic neither. All are absorptive and decomposers. Wrong. All fungi are not that bad, because some fungi are forming symbiotic association, mycorrhizae, or maybe people in lichens, they live together with the others. So they don't have to be beggars like decomposers. Neither. Fungi don't produce spores with flagella. Fungi don't produce spores with flagella. No, that's wrong. Why? Chytridiomycota flagellated stages are there. Wrong statement. It was a sexual reproduction. Ascomycota produce two types of conidiospores. In sexual reproduction, Ascomycotas produce two types of conidiospores. What do you say about that? In sexual reproduction, Ascomycota produce two different types of conidiospores. Sexual reproduction, conidiospores. Are you mad? Neither. Wrong, neither. Basidiospores of Basidiomycota are exogenous. Basidiospores of Basidiomycota are exogenous. That's correct. Exogenous is correct. The answer is 5. That is absolutely correct. Moving to question number 13. Question number 13. Which one of the following is not a characteristic feature of Arthropoda? Which of the following is not a characteristic feature of Arthropoda? Presence of book lungs. Presence of book lungs. Presence of book lungs. Arthropoda. Book lungs. What do you say about that? Do arthropods have book lungs? Lamai. Arthropods lutter book lungs ema thiyana wada mokada kiyanne? Yes, Lou. Yuthika says yes. Kahira says yes. Enang Hari. The presence of calcareous exoskeleton. Calcareous exoskeleton. Who has calcareous exoskeleton? Who's having calcareous exoskeleton? Arthropoda. Presence of tracheal system, correct. And then presence of Malpighian tubules. Malpighian tubules, Arthropoda, do they have it? We discussed that today. Yes, they have. Presence of segmented solid nerve cord. Segmented solid nerve cord in Arthropoda? Yes, correct. So what's your answer? What is your answer? People, I have to select the second one. Calcareous exoskeleton. We'll take it for mollusca. We'll take that for mollusca. So how about the second one taking as the answer? Question number 14. Question number 14. The cells are alive at maturity and provide support in the plants are present in. What is that? Cells that are alive. Cells that are alive at maturity and provide support to the plant are present in. Which one is that? Which one is that? Epidermis, vascular tissues, sieve tube elements, cholenchyma. We will take cholenchyma. Cholenchyma is the answer. Oh, Kolenkaimar is the answer. Moving to question number 15. In which portion co-cambium originate in the roots during secondary growth? In which portion the co-cambium originate? Kohen the lamai co-cambium originate when they pericycle, endodermis, cortex, secondary phloem and primary phloem. What is the answer? Co-cambium. Co-cambium is the pericycle. Remember that pericycle form the vascular cambium and the co-cambium both in the root. Can you remember that? Neither. So therefore the answer has to be pericycle. Answer has to be pericycle. So we are taking first one as the answer. Question number 16. Which of the following would happen when turgid cell is placed in solution which has similar solute potential to the cell? Which of the following would happen when the turgid cell is placed in a solution which has similar solute potential to the cell? Which has similar solute potential to the cell? Water potential question. Water potential question. Come there. Water potential question. Which of the following would happen when the turgid cell is, what do you call a turgid cell? Fat inflated cell. The turgid cell is placed in similar solute potential to the cell. So nothing happens. No endosmosis, no exosmosis. Why, people? Because it's now in isotonic solution. Right? What happens if you work with another student who's having the similar knowledge like you? Nothing happens. If you work with what do you call bright case, you improve. If you work with a dumb person, you also dumb. You also become a little bit dumb. He improves and you become dumb. Right? When you work with an intelligent person, that intelligent one become little dumb, but you shine up little bit. So good method. Find some good friend. Exosmosis until. Which of the following happen if the turgid cell is placed in a solution which has similar solute potential to the cell? Which has similar solute potential to the cell. That we have to think about a little bit. Similar solute potential to the cell. Now your cell is turgid. Your cell is turgid. Your cell is turgid. This is your cell. Turgid. So this turgidity, so if you write the water potential for this cell, when you write water potential psi S plus psi P, this psi P is positive. Again turgid. Outer solution solute potential is similar to the cell solute potential. This is the cell solute potential. Now my question is, which one is having more water potential? Which one has more water potential, outer solution or your cell? Which one has more water potential? Which one has more water potential? Come on, think and answer my question. Outer solution solute potential and the water potential both are equal. Outer solution solute potential is its water potential. Water potential, this is solution. Water potential of the solution and the solute potential of the outer solution is same. Right? Now, if you take, let's take that this is minus 2 and this one also minus 2. Right? But there is a plus value. This one, let's take it as plus 1 kilogram. Plus 1. So what is the water potential here? Water potential here is minus 1. Why you have plus 1? And I make a minus 1. What is your outer solution water potential? Minus 2. So who's having more water potential? Cell has more water potential. Am I correct? So work it out, people. Here the cell solute potential and the solute potential of the outer solution, they are same, minus 2, minus 2. So that cell water solution water potential is also minus 2. Harine. But cell water potential is now, if you assume here this one as plus 1, because it turgid ne plus 1, then the cell water potential becomes, people, minus 1. Cell water potential is minus 1. Outer solution water potential is minus 2. Which has more water potential? Cell has more water potential. So from the cell, water now moves out. From the cell, water comes out. So what do you call that? Exoosmosis. Please raise hand if you understood that. Please raise your hand up if you understood that. Raise your hand up if you understood that. I have one person. Sabik Mohammed Azmat Venukta Thari Neda. Arenena fit Neda. What should be the answer then? What should be the answer? Answer is, people, exosmosis until water potential equals to the water potential of the solution. That is correct. So the answer is going to be 2. Answer is going to be 2. Very good. If you still can remember some water potential, the things better, right? I promise you that I'm going to take water potential discussion. I'll take. Don't worry, right? Question number 17, my dear. The elements essential for the components of middle lamella. Elements essential for the components of the middle lamella, cofactor of the enzyme, and activate the formation of chlorophyll in the plant, correct sequence. Maybe, people, middle lamella component nang calcium, nether, and cofactor of the enzyme, calcium, cofactor of enzyme, and activator for the formation of chlorophyll. Activator for the formation of chlorophyll. Cofactors for enzyme. Enzyme cofactors. Dear, I can't remember, dear. What is your answer? What is your answer for that? I think I need to check that. Calcium comes first in everything. You can't take the third one and the fifth one. So calcium first, second, third. And then people, cofactor of enzyme. Cofac. Enzac. Cofac. Enzac. Cofac. Potassium. Cofactor. Check it. Am I correct? Potassium. And then chlorophyll. Activate the formation of chlorophyll in plants. Activate. Enzyme activate the formation of chlorophyll. What is your answer, Lamai? You want me to check that? What is your answer? What is your answer? You didn't tell me the answer now. Zinc, it says activator of many enzymes of chlorophyll. Zinc is okay. Potassium balloon hari. Cofactor of hari. Answer is the second one. The given marking scheme given answer is okay. Question number 18, people. In Seekers, the structure of ovule which nourishes the developing embryo. Structure of the ovule which nourishes the embryo. In Seekers, the structure of the ovule which nourishes the embryo is the female gametophyte. We'll take this one as the answer. Question number 19. Question number 19. Formation from the given combination of plant growth regulators regarding apical dominance, which act antagonistically. Auxin gibberellins, auxin cytokinin, auxin abscisic acid, Auxin and abscisic acid. Abscisic acid, what we said people, not abscission in a leaf senescence. Auxins, auxin I remember like abscission. Stop abscission kila hogeta mana mataka. Stop abscission. Plant hormones. Plant hormones. Ayyo. Plant hormones. Hormones. Plant hormones. Auxin retard abscission කියලා තියෙන්නේ. එතකොට we can't take the marking scheme answer ගන්න බෑ නේද? Auxin and abscission wrong. So, abscisic acid and ethylene ගන්න බෑ. Auxin and cytokinin. From the combination of plant growth regulators regarding the apical dominance which act antagonistically. Auxin and gibberellins. Wrong neda. Auxin and cytokinins. That is correct. Ne? Apical dominance and inhibiting the apical dominance. So, second is okay. Ne? 19th people, මොකද්ද මම දුන්න answer එක 4th කියලා. නම් වැරදි නේද? Second කියලා ගන්න ලවයි. Second 19th second හරිද? මොකද asking plant growth regulators regarding apical dominance. So anyway, එතකොට auxin and cytokinin auxin promote the apical dominance, cytokinin stops it. So answer has to be second, my dear. Answer has to be second. So I'm modifying my answer. Second one is the answer. Second one is the answer. Right, moving to page number 20. Page number 20. Page number 20. Not page number 20, question number 20. Ne? Question number 20. Question number 20. Hari lamai. This time there is a problem in the printing. Ne? Upside down. Ne? Upside down. Ne? And question number 20. Which of the following statement regarding plant response to the internal and external stimuli is correct? To the internal and external stimuli is correct. Cryptochrome is a major blue light photoreceptor. Which of the following statement regarding plant response to internal and external stimuli is correct? Cryptochrome is a major blue light photoreceptor. Cryptochrome, blue light photoreceptor, correct. Increase far red to red ratio inhibit branching. Increase far red to red ratio inhibit branching. Now resource book they have given this wrong. එතකොට අපි තීන විදිහට ගමු නේද? We'll take as the way it is given, right? Exposure to direct sunlight and the proportion far red to red light thereby stimulate branching. Far red to red inhibit නෙවෙයි thereby stimulate branching කියලා තියෙන. එහෙනම් අපි inhibit branching wrong කියලා ගමු. එහෙනම් wrong. First is okay වගේ. Plant detect gravity by settling statolith. Plant detect gravity by settling statolith. Plants detect gravity by settling statolith. That is correct, ne? Right? Plant detect gravity by settling statolith. Correct, ne? The statoliths are present in the root. Statoliths are found in the cell elongation zone of the plant root. No, the root cap, wrong. Red light photoreceptors are reason for the slowing down of hypocortical elongation. Red light photoreceptors are the reason for slowing down of hypocortical elongation. Red light photoreceptors are the reason for slowing down of hypocortical. And the blue light photoreceptors, wrong. So I'll take, what is your answer? What is your answer for that question, Lamai? What did you take? I'll take third one as correct. Cryptochrome is, this is also correct. First and third. First and third wage mata inni lamai. first and third කියලා. හරිද? අනික් ඒවා wrong නේද? අනිත් ඒවා wrong. Right, first and third. What is your answer? What did you take? චතුූප. මොකද ගත්ත උත්තරේ? First and third. I took second. Second ගන්න බෑනේ ළමයි. Increase far red to red ratio inhibit කියන්නේ දැන් resource book එකේ තියෙන්නේ stimulate කියලා. That's wrong, but there is no point of arguing that. Ne? So we'll take it as the way it is given. Ne? Question number 21, lamai. Which of the following event occurring during cellular respiration is correct? Which of the following event occur during cellular respiration is correct? Series of protein and non-protein molecules involving in movement of electrons. Series of protein and non-protein involves in moving of electrons. Series of protein and non-protein molecules involving in series of protein and non-protein molecules involving in moving of electrons. Hari. Correct. Not all the cells do glycolysis. No, wrong. Neither. All cells do glycolysis. Neither. Because it's something common to both aerobic respiration and anaerobic respiration. And then, correct. Series of protein and non-protein molecules involving in movement of electrons. Correct. Not all cells do glycolysis. Not all cells do glycolysis. Wrong. In anaerobic respiration, regeneration of NAD plus occurs. In anaerobic respiration, regeneration of NAD occurs. Correct. Finally, hydrogen acceptors of lactic acid fermentation is 2-carbon compound. Lactic acid fermentation is 2-carbon compound. Wrong. It is pyruvate, 3-carbon. Calvin cycle occurs in the matrix of the mitochondria. Wrong. Neither. Krebs cycle occurs there. Ne? So, A and C, lamai. A and C. I'm taking answers as A and C. Marking scheme answer, you have to correct AC only. Marking scheme answer, AC only. Question number 5. Question number 5. Question number 22. Ne? Sorry, question number 22. The correct statements about the history of the evolution and classification is are. The correct statements about the history of classification is are. Ancestors of arthropods, chordates and other animal phyla originated 700 million years ago. Ancestors of arthropods, chordates and other animals originated 700 million years ago. Ancestors of arthropods, chordates and other animals, ancestors of arthropods, chordates, 670 ne lamai. 670 ne wrong ne. Major radiation of mammals, birds and pollinating insects occurred during the Cenozoic. Major radiation of mammals, birds and pollinating insects. Cenozoic correct. So I'll take this one. Ancestors of arthropods wrong. This is correct. Then species is the smallest group of individuals that share common ancestor. Correct. Neo-Darwinism generally denotes the integration of Mendelian genetics and population genetics. Neo-Darwinism generally denotes integration of Mendelian genetics and the population genetics. Neo-Darwinism generally denotes integration of Mendelian genetics and population genetics. Wrong Nedalamai. Why Neo-Darwinism has 3 parts? What are those 3 parts of the Neo-Darwinism? Darwin's theory of natural selection, Mendelian genetics, and then population genetics. Ne? Right? I'll take it as wrong. I'll take it as wrong. Right? And then the origin of the human species took place 6 to 7 million years ago. Wrong, neither. 195,000 years. Ne? So B and C. B and C must be the answer for that. Question number 23. Characters distinguish an angiosperm from other terrestrial plants. Characters that distinguishes the angiosperm from the other terrestrial plants. Double fertilization. Characters that distinguishes from double fertilization. Other terrestrial plants. Yeah, angiosperms have double fertilization. Neither. They fertilize 2 times. Neither. Double fertilization. Process of embryo sac. Correct. Development of pollen tube. No, pollen tube. What do you say? Development of pollen tube. No, neither. Coccus has pollen tube too. Wrong. Gametophyte non-dependent on the sporophyte. Gametophyte non-dependent on the sporophyte. Gametophyte non-dependent. Wrong. Neither dependent. Neither lamai. Wrong. Presence of endosperm. No, endosperm is present in coccus also. So AB must be the answer. Question number 24. Features of free-living forms of phylum Platyhelminthes that cannot be seen in parasitic forms. Features free-living forms of phylum Platyhelminthes that cannot be seen in the parasitic forms. Parasitic forms don't have cilia and sensory structures. Will take these 2 as the answer. C and D lamai. C and D. Bisexuality. We didn't say anything neither. Larval forms cannot be seen. Larval forms are present in the parasitic forms. You can't take B. Cross-fertilization. Parasitic forms. Cross-fertilization. What they mention about the parasitic forms, the cross-fertilization, parasitic forms. Let me check that, people. The memory is missing in that part. Parasitic forms, Platyhelminthes, ne? It says separate sexes, nitrogenous. Some shows asexual reproduction, bisexual, but show cross-fertilization which is internal. Bisexual shows cross-fertilization which is internal in parasitic forms. There are several larvae stages, direct development of free-living examples without a larva stage. It says cross-fertilization which is internal. Some shows asexual reproduction by regeneration, bisexual but shows cross-fertilization. What this one says? Feature of free-living forms of Platyhelminthes cannot be seen in parasitic forms. They have free-living examples have cilia for the locomotion. Some shows asexual reproduction by regeneration, bisexual but shows cross-fertilization. Bisexual but shows cross-fertilization. So we'll take wrong neither. CD as the answer. Free-living forms, right? So we'll take CD. Question number 25. Correct regarding phloem translocation. Loading always is an active process, correct? And then forces the sap under the negative pressure. Forces the sap under the negative pressure. And in the positive pressure, wrong one. Phloem sap flows under positive pressure. We saw the evidence when you tap the rubber tree and all, the phloem sap flows out. Sinks usually receives sugar from the nearest sugar source. And then phloem sap can be transported in opposite direction. 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 Opposite direction. Possible, Nedlamai? Correct, Ned? Correct. And then water cannot move by osmosis. Water cannot move by osmosis. No, water moves by osmosis. So A and D. Sinks usually receive sugar from the nearest sugar source. Wrong, neither. A and D lamai. A and D must be the answer. So marking scheme answer again I am modifying. So it has to be last one A N D. A N D, not A C D. A N D, ne? A N D. Right, moving people for the structured part. Moving to the structured part. Find the structured part. Moving to the structured part. Right, moving to the structured part. Part A, structured essay. What are major characteristics that distinguishes living from non-living? You can write that, no issue. Order, organization, metabolism, growth and development. We'll take that part. Correct. Right, next one, people. What are the most abundant organic compound on earth? Carbohydrates. Carbohydrates. One of you asked a question the last time in the practical class whether you can take. What did you ask? Sugars kila devi. Sugars the carbohydrate I think better. So C is correct. Check in the resource book now. Mokadda C statement. What is that? 20th C statement. C statement correctly. It says since you should receive sugar from the nearest sugar source. Sinks usually receive sugar from the nearest sugar source. Source and the sink at two different locations. Sinks usually receive sugar from the nearest. Sink is the one which stores it. Nearest sugar source. No, I thought it like the source is at a distance and then it transport. Anyway, we'll check. If that sentence is given. Page number, can you tell me the page number, Tahira? What's the page number? 133. Phloems at moves. Sinks usually receive sugar from the nearest sugar source. For each sieve tube, the direction of transport depends on the location of the sugar source and the sugar sink that connected by the tube. Therefore, neighboring sieve tubes may carry sap in opposite direction if they originate and end in different locations. Sinks usually receive sugars from the nearest sugar source. Then we'll take that as correct. Then the marking scheme answer is correct. Sinks usually receive sugar from the nearest sugar source. For each sieve tube, the direction of the transport. Because I thought like the sink and the source are distantly located, right? So that they have to transport it to a distance. So anyway, I would like to correct them my. Then I have to fix this. Then ACD. Then ACD. Right, moving people, moving to these structured part. Structured part. Write down three main most simple form of above mentioned compound. Carbohydrate in living body. Write down the three main functions of the most simple form of above mentioned. Again, sugars, sugars, energy source, building blocks and all. Right? So we'll take that part. Name two major nitrogen containing polymeric compounds in living organisms and indicate two major function of each. Compound protein, catalyze biochemical reactions. Nucleic acids store and transmit genetic information, like that you can take. Draw a label diagram of the mitochondrion. I have given marks people for this. See whether you have labeled DNA, outer membrane, inner membrane. Those details you need to label them, right? And so these points must be there: DNA, outer membrane, inner membrane, cristae, these things. Okay. State two major functions of mitochondria in eukaryotic cell: synthesis of ATP in aerobic respiration, Name the structure of an animal cell which carry out following functions: detoxification of toxic compounds during cellular. Name the structure of an animal cell which carry out following function: detoxification of toxins formed during cellular metabolism, peroxisome, and then digestion of worn out organelles, lysosomes. State 2, structures found in the plant cell which are not found in typical animal cell: glyoxysomes. State 2, structures found in plant cell wall which are not found in animal cell: cell wall, chloroplast. Simple, right? What is an enzyme? Macromolecule which act as biological catalyst. What is the basic function of an enzyme in the catalyst of biological reaction? Lower the activation energy. And then state 4 factors affecting rate of enzyme reaction. These are the 4 factors. I think it's simple. Explain the term specificity. Enzymes catalyze only specific type of reaction by binding with specific substrates. Second question, people, name the generic name of the heterosporous seedless plant with vascular tissue. Selaginella. Generic name, heterosporous, seedless, vascular. So you have to underline, okay? If you don't underline, people, then you don't get marks. What is the purpose of the keys in studying group organisms and identify them? And then what is the commonly one? Dichotomous key. And here a dichotomous key is given. So how did you fill that dichotomous key? It says, people, now you have to follow the list neither. Use the dichotomous key to identify 5 leaves in the picture. Leaf is oval shaped. Leaf is oval shaped. This is oval shaped. This is oval shaped. This is oval shaped. So A, B, D. Leaf is oval shaped. A, B, D. A, B, D. Leaf is star shaped. C and E. C and E. So go to step number 2. Leaf is shorter and wide. Shorter and wide, which one? A and B. Shorter and wide, A, B. Leaf is longer and narrow, then that is D. Hickory. So D is hickory. 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 D is hickory. Leaf has smooth edges. Smooth edge is B. B. Again is sweet gum. Sweet gum. And leaf has jagged edges, maple. That is Mukallamai. A neda. A is the maple. Sugar maple. Sugar maple. It must say, go to people, leaf has 4 veins in each side. 4 veins in each side. Breech. Wait. Now, leaf has star shape, go to 3. Right, we go to, oh. I made a mistake. Oof. I made a mistake. I will start once again, over again. Leaf has shorter and wide, go to 4 kilocan. Go to 4. This is A and B. This is D, right? That part is okay. Go to 4. Leaf has 4 veins on each side. 1, 2, 3, 4. 4 veins, breech. 4 veins, breech. 4 veins, hickory is okay. 4 veins on each side, breech. 4 veins on each side, breech. 1, 2, 3, 4, breech. Again, A, breech. A, this is A. 5 veins on each side. 5 veins on each side. 5 veins on each side is B. 4 veins on each side, breech. 5 veins beach. ඊට පස්සේ the step 3 smooth edges sweet gum. C නේද? C sweet gum. C and C. What is C, people? Sweet gum. Sweet gum. And then the next one, either sugar maple is E. Sugar maple. Right, sugar maple is that. Right? Okay, is that okay, people? Hari neda. So E sugar maple. Hari ne. Then we move to next part. The high metabolic rate. Write one main physiological adaptation. Write one main physiological adaptation shown by class Aves high metabolic rate. We'll take that as correct. Name two external structural features present in the class Aves not present in reptilia. Presence of keratinized feathers, beak without teeth. It is believed that the terrestrial plants were evolved from aquatic plants such as green algae, and bryophytes are considered as the first group of plants adapted for the terrestrial environment. What are the evolutionary advanced characters shown by bryophyta which are not seen in green algae? Now you can write that, neither. What are you going to write, people? What are you going to write? These things. Wold spores, multicellular gametangia, apical meristem, and dependent embryo. What are the difference between dispersal of the spores and the gametes in Neplephis? Neplephis spores by wind, gametes by external water. Hurry. It was a name the plant genera which shows following characteristics. External water is required for fertilization. Pogonatum, Neplephis, Selaginella. Possess non-motile sperms, Netum and Pinus. Non-motile sperms. Right? Plant Anthophyta. How about Anthophyta? Anthophyta don't have a sperm, the sperm nucleus only. And then having free and independent gametophyte, Pogonatum and Neplephis. Heterospory, Netum, Pinus and Caecus. Heterospory, Netum, Pinus, Caecus. You can write Anthophyta also. Is there a list given before? Is there a particular list given for that? Is there a list given? Heterospory. Name the plant genera which shows following characteristic. Plant genera, genus you are right, right. So then we'll forget about Anthophyta. Or if you're writing genera, what are you going to write, people? We didn't learn any genera for Anthophyta, did we? So we'll just leave them. Showing secondary thickening, secondary growth, Netum, Pinus and Caecus. Oops. Showing secondary thickening, Netum, Pinus and Caecus. All you have to underline. If you are not underlining, people, there is a chance of losing marks. Don't do that. Indicate the stages of the following life cycle in which the meiosis take place. Agaricus, Basidium, Caecus, Megaspore mother cell, Neplephis inside the sporangium. Right? I took them. What is plant growth? What is plant growth? Irreversible increase of dry mass associated with the development of plants. Draw a diagram to show primary structure of the monocot and the dicot root. So I'm accepting these two diagrams for the primary structure, right? And then people describe the formation of vascular cambium of the stem. Stem vascular cambium is formed by ring of vascular tissues produced between the primary xylem and the primary phloem. Form undifferentiated cells of single cell layer often outside the pith and the primary xylem and inside of the cortex and the primary phloem. This you have to write. It was describe how it is formed in the root. Root vascular cambium formed laterally exterior to the primary xylem and interior to the primary phloem. That is given in the resource book. Then weaved a cross section. Vascular cambium appears as a ring of initials. Describe the structural and the functional difference among those initials. Vascular cambium has two initials. One is involving in forming the xylem and the phloem, and the others are involving in forming the xylem vascular race. Others are involving in vascular race. Elongated and oriented in their long axis, which forms the vascular race, and shorter and orient perpendicular. Elongated ones are forming, elongated ones are forming vascular tissues. Perpendicular ones are forming the race. Produce tracheids, that is correct. Companion cells. What is the question? Weave a cross section. Vascular cambium appears as. Describe the structural and functional difference among those initials. Now, these perpendicular ones are forming vascular race. Given, okay. Given vascular race. Forming vascular race, produce vascular race. That is correct. Right, people, moving to next page. That is the essay questions. That is to discuss essay questions. What is a vital inorganic molecule? Existence of life on planet. Mention two reasons. Now, people, I saw many students have described this. Right? It says mention. Now, you have to check these keywords, people. Mention. You just mention it. You don't have to describe it. So, unnecessarily, you don't have to describe it. Right? Then people describe the major properties of water that are important. So that most of the students have written this part correctly. Right? There is no issue with that. Right? But the writing those basic properties, the properties of water, and then describing the cohesive behavior, so on. You have to go on describing. So versatility as a solvent, and those things you can describe. Now, people, second one, describe the xylem tissue of netum. Now, xylem tissue of netum, I expected you to write the netum is a plant with xylem vessel elements. I just wanted you to pick that point. It's having the xylem with the xylem vessel elements, and then describe people the xylem vessel element details which is given in the plant form and function part. Xylem vessel element, tracheids, and that description with the diagram I expected. Now, some students have written 2-3 sentences which is given in the divised lesson. That's not sufficient, okay? So, I expected a full answer here with some diagrams. I expected you to draw the xylem vessel elements and all, right? So, that part I think some students fail to answer, okay? And then part B, describe the translocation of photosynthetic products from leaves of mango plant to the fruit. Now, this is regarding the phloem transport, right? This is about the phloem transport, right? I ask students to describe the translocation process. Things are usually receiving sugar from the nearest sugar source. The direction of transport depends on the location of the, this one, I missed that. Here also it's there. This one. Okay. The direction and the transport depends on the location of the sugar source and sugar sinks connected by the tube. The first step in translocation is transport loading into the C tube element. Phloem loading occur. Phloem loading occur, and then endosmosis take place. The pressure inside the C tube element near the source is going to increase. This pressure is now creating a pressure gradient. Along the pressure gradient from along the C tube, we can see that the flow occur. The first step in translocation is the phloem loading, and in some species it moves from mesophyll cells to the C tube element via symplast. Many plant sugar moves into the phloem require active transportation. Sucrose is more concentrated in the C tube element and companion cells than in mesophyll cells. Sucrose is unloaded at the sink. So you have to follow this order, right? However, at the concentration of free sugar in the sink is always lesser than the sugar content during the growth and metabolism of the cells. Sink or converted into insoluble polymers such as starch. Right? From the sink, sugar is stored and then removed. As a result, sink sugar level is less compared to the C tube element. That create a gradient, and then along the gradient, this movement occur. Sugar molecules diffuse from the phloem into the sink. So this part. Then people in the third section, you have to describe, you have to write a short note. So when I give you questions, people, right, you can select and write 2 essay questions. So galls of plants. We learned that in the cell cycle lesson, galls is occur by uncontrolled cell division. The plant cell division is controlled by the proper balance between plant growth regulators such as auxin and cytokinin. When the balance is lost, we can see that the undifferentiated cell mass can be produced, which we call as the gall. That develop different parts of the plant, they can develop, and sometimes they are formed by after infecting with unique organism. We call them as a gall causes: virus, fungi, bacteria, insects. And what else? Insects and mites can cause galls. Usually gall cause they penetrate into the plant. Gall causes. So these are the gall causes and develop an abnormal growth. Right, second one, people, about the radial transport. Radial transport. How radial transportation take place. Right. We learn radial transport is the transport of water from the cortex into the vascular cylinder. Transport of minerals and water from the soil to the root cortex into the xylem of the root is called as the radial transportation. And the roots are used, 3 roots are used: apoplastic, symplastic and the transmembrane root. And then water and the solutes are move along the continuum of the cell wall. The extracellular space is known as the apoplastic route. Epidermal mist blocks the apoplastic route and keep unwanted toxic materials out. The endodermis also prevents solute that have accumulated in the xylem from leaking back into the soil solution, andoni. And then symplastic route, water and the solutes move along the continuum of the cytosol, and then substances can move from cell to cell via plasmodesmata. Some substances can move more than one route. The least resistant for the transport is found in the apoplastic route. Therefore, more water use apoplastic route. Both the diffusion and active transport involves in transport. When water and the minerals can enter to the trachea and the vessel elements, the transport to the shoot system by bulk flow only through the apoplast. Right. Ilan lamai significance of C4 photosynthesis kila question එක importance. So we learn 4 importance. The C4 pathway helps to improve the efficiency of carbon dioxide fixation at lower carbon dioxide concentration, prevent the gateways of photorespiration in hot dry climate, and then this produces carbon dioxide, this reduces carbon dioxide intake At low carbon dioxide concentration, C4 mechanism increase the efficiency of photosynthesis by C4 plant exhibit better water usage and other nitrogen usage. They can acquire enough carbon dioxide even when keeping their stomata closed. Since less rubisco is needed, they have better nitrogen use efficiency. So those are the parts, right? So people, that's all then for the day. Paper number 14, we'll go with next paper, paper number 15, I'll give you on Saturday. But in the meantime, I want to take a small discussion on water potential also. If I discuss water potential, people, we won't be able to do anything else. So shall I give you a water potential, some question tut, people? Better need. I'll give you some water potential calculation tut rather than giving anything else on coming Saturday. Then next Wednesday we'll discuss a little bit of water potential, and if time, yeah, we'll do that, right? Okay then, that's all for the day. Thank you very much for your participation. Hari enang api will meet people on another day, right? Okay then, see you all. Bye bye. Good night.